Dear students, today talk on organs of immune system. It involves primary lymphoid organs and secondary lymphoid organs. I will take primary lymphoid organs. The primary lymphoid organs are the major sites of lymphocyte development, that means lymphopoiesis. Lymphocytes differentiate from lymphoid stem cells, proliferate and mature into functional cells called immunocompetent cells. In mammals, B cell maturation occurs in the bone marrow and T cell maturation occurs in the thymus. I will take thymus. The location of thymus is located in the thoracic cavity, in the mediastinum, just above the heart and beneath the breastbone. Next to know origin of thymus. In mammals, thymus develops from the endoderm of the third and fourth pharyngeal pouch. Structure about thymus. The thymus is flat, bilobed, grayish lymphoepithelial organ. Each lobe is made up of lobules separated from each other by strands of connective tissue trabeculae and covered by a capsule. Each lobule consists of two compartments. The outer compartment is the cortex, which is densely packed with immature T cells, the thymocytes. And the inner compartment is the medulla, which is sparsely populated with the mature thymocytes, which express CD44, not found in the cortical thymocytes. The diagram gives certain important parts. The thymus mainly consists of two regions, cortex and medulla. It consists of certain important parts like trabeculae, capsule, thymocyte, nerve cells, dead cells, dividing thymocytes, interdigitating dendritic cell, macrophages, blood vessels, cortical epithelial cells, Russell's bodies, etc. Second one is the bone marrow. Bone marrow is also primary lymphoid organs. The location of bone marrow. Bone marrow is found in the cavities of most bones in the body, including the skull, ribs, sternum, femur, and spine. In birds, no bone marrow is found to be present. Instead, a lymphoid organ named bursa, identified by Fabricius, called bursa of Fabricius, is present and performs the same duty or same function of bone marrow of mammals. So instead of bone marrow, the bursa fabriciae functions as bone marrow in birds. Structure of bone marrow. The bone marrow of different bones mainly consists of a sponge like a reticular framework located between long trabeculae. So between the trabeculae, there is a sponge-like reticular framework. The spaces in this framework are filled with fat cells, stromal fibroblasts, and precursors of blood cells. All the blood cells originate from the common stem cells. It lacks markers of differentiated blood cells and instead expresses two proteins called CD34 and stem cell antigen 1. These markers are used to identify and enrich stem cells from suspensions of bone marrow or peripheral blood cells for use in bone marrow transplantation. The pluripotent stem cells become committed to differentiate along particular lineages. That means erythroid, megakaryocytes, granulocytic, monocytic, and lymphocytic by the stimulation of cytokines. Colony stimulating factors, which are produced by stromal cells and macrophages in the bone marrow. So instead of bone marrow, there is a bursa fabricis in birds. The bursa fabricis is an ovoid sac-like structure measuring 3 centimeter in length and weighing 3 grams at the maximal development in chicken. The bursal lumen is connected to the last cloacal chamber, the proctodeum, by a short duct. Anatomically, bursa comprises a central, highly staining medulla and a dark staining peripheral cortex. So, cortex and medulla part. Besides the reticular and epithelial cells, lymphocytes, plasma cells, macrophages, and some granulocytes can be found in the bursal follicles. The size of the bursa gradually diminishes at the adult age, around 4 months in case of chicken, due to rise of level of circulating steroidal sex hormones like testosterone and estrogen. Bursa develops as an outgrowth from the endodermal epithelium of the intestine in the cloacal region on the 10th day of incubation and forms a localized swelling forming follicles. Large numbers of lymphocytes can be found around them and at the 17th day of incubation. The formation is completed. So the diagram shows vertebral column, bursa fabricis, bursa duct, cloacal aperture and gut. So bursa fabricis functions as bone marrow in mammals. So bone marrow of mammals going to differentiate and mature the B cells, B lymphocytes. So B lymphocytes are differentiated and matured by bone marrow, but in case of birds, in case of chicken, instead of bone marrow, there is a bursa fabricis. In the bursa fabricis, the B lymphocytes are differentiated and matured. For any clarification, for any doubts, send me the messages. Thank you.